Good evening to Sir Fuad and my fellow friends. So I'm from Faculty of Health Science, Department of Environmental Health, Bachelor of Environmental Health and Safety. So my name is Fendi bin Mita. My metric number is BEHP 24046200, cohort 23, and I'm currently conducting training at Pejabat Kesihatan Kawasan Sandakan. So my presentation today is about practical training for water quality control. So the KEMAM, or known as Drinking Water Quality Control Program, uh, was introduced by the Engineering Service Division, Ministry of Health Malaysia in 1983. So this comprehensive program monitors the quality of drinking water from public water supply system through field and laboratory analysis to ensure safe drinking water for all. So this is my logbook activity 5.1.1, which is prepare sampling schedule and taking sample for microbiological and chemical analysis. So this report documents a comprehensive water sampling activity which is conducted at the Segaliud Water Treatment Plant on 14 July 2025. So all the procedures followed the MS 1850 Part 3 2005 MOH protocol and also internal guideline. Alright, so this is the sampling objectives and also the methodology. The primary objective is to perform in situ analysis of key physical parameters, to collect water samples for chemical, heavy metal, and bacteriological analysis, and also demonstrate proper sampling technique to junior personnel, and also verify efficiency of cold chain handling and sample documentation. So we have parameter group 1, group 2, 3, 4, and 5, which is physical parameters, chemical parameters, heavy metals, bacteriological parameters, and also additional parameters if needed. So the sampling activity aims to assess the overall quality of water at various stages of treatment and distribution. So ensuring compliance with stabilized water quality standards. This is the sampling schedule, or known as sampling frequency schedule. So this is our sampling point. We have raw water treatment plant outlet, or TPO. We have SRO, service reservoir outlet, and also auxiliary. All right. For group one, which is physical group, we take the sample weekly for all for sampling point. For group 2, which is chemical, we do monthly for raw water, for treatment water, treatment plant outlet, and also service reservoir outlet. But for auxiliary, we do two times a year. So for group 3, which is heavy metals, for raw and TPO, we take yearly, four times a year. And for the TPO, maybe two times a year. Non at SRO, and also we do yearly at the auxiliary. For group Pro, which is bacteriological, we do four times a year at raw water and also treatment plant outlet. And then whenever necessary at the SRO and also at the auxiliary. For group 5, we take whenever necessary for all four at the sampling point. So this is the management of drinking water sampling activities. So we have preparation, we have field sampling, quality assurance, and also sample handling lah. Okay, so this is the essential field equipment. We must have sterile plastic sample containers, bag for water sample. We must have preservatives, alcohol and sampling equipment, color label for sample identification. We need to bring form S1, S2, S3, and S4 as relevant. And also plastic bag for chemical sample bottle, and clean sample shipping containers with pre-frozen ice pack, and temperature reference bottle, and also equipment for physical parameter field test. So first, we need to know what is the purpose of water sample preservation. So the main purpose uh, of preserving water sample is to maintain the original condition by stabilizing important parameters that are subject to change. So this is crucial because this process minimizes chemical alteration that could occur between the time of sample collection and also and laboratory analysis. So by preserving the samples effectively, the result obtained from the laboratory testing will better reflect the true composition of the water at the moment it was collected. So sample preservation is very important for the sampling activities. So we have a few water sample preservation preservation methods, which is first one is pH control with acid. We can add acid for meta analysis, and acid form chemical bond, which is stop the reaction, minimize precipitation of dissolved ions, and reduce volatilization. We also use opaque bottle, which is amber or opaque bottle that help to reduce reaction rates due to light exposure and inhibit microorganism reproduction by protecting samples from sunlight. And then we have temperature control. We need to cool the sample to 0 until 6 degrees Celsius to inhibit microorganism reproduction and also minimize volatilization and biodegradation and reduce reaction rates. We also have other additional preservation methods, which is we use sodium thiosulfate uh, added to the what added to the treated water sample for bacterial testing to deactivate or neutralize pre-chlorine and maintain the bacterial population. We also have sterile containers. We write on yellow tape for raw water entire bag, white tape with the sodium thiosulfate for treated water to avoid external bacterial contamination. So we also fill it to the brim. So water sample are filled completely to avoid losing volatile parameter like mercury and trihalomethanes. So this is acid preservative concentration. Okay, so you can take a look at this to see the bottle, type of bottle, what are the parameter, what are the preservative needed, and what are the label color of the bottle or our container, what are the temperature, and how long we can hold it. So this is the material and equipment for chemical analysis. We have container, which is clean acid-wash polystyrene or glass sampling bottles. Okay, the size are vary depending on the parameter being tested lah. So we also have preservative, nitric acid for trace metal, hydrochloric acid. This one is optional depending on the analysis, and we must have storage and transport, which is ice pack and cooler box, plastic bag for sample bottle. So this is the picture of sampling activities at the Scalia water treatment plant. So the first picture is field team conducting the in situ water quality testing. The second one is sample collected activities following proper protocol to ensure the representative sample, and the sample preservation using appropriate chemical preservatives to maintain sample integrity. And others are sampling activities and also documentation activities. So this one is chemical sampling bottles. We have bottle A and bottle B as shown. This used for both raw and treated water. And this is additional chemical sampling bottles. So we have bottle C and bottle D. Okay, used for raw water only for C, but for bottle D is used for both raw and treated water. We also have specialized chemical sampling bottle. This is special for ammonia. It's 3 and nitrate and nitrate analysis. This is used for both raw and treated water. So this is our sampling activities. This is the briefing to enhance the understanding of water supply system. Okay, next we go back. Water sampling from tap for analysis for physical parameters, group 1. So first we need to clean the faucet to remove dirt. Then we need to let the water run for 2 until 5 minutes to remove the deposit from the pipe. So for pipe that are less used, we need to open it until 5 to 10 minutes. Same with the low water pressure pipe. Yeah, so make sure the water flow in without touching the edge of the bottle mouth to prevent contamination, and also fill the water sample into the bottle with slow flow to avoid turbulence and air bubbles. We need to perform analysis of physical parameter pH, residual chlorine, turbidity, color, DO, conductivity, and total dissolved solids using a spot test tool immediately in the field, and record the remaining chlorine, pH, turbidity, and color reading in the form SA1 and SAB. So this is the flow chart of water sampling bacteria at the sampling station. First, we need to remove all connection at the mouth of the faucet. Use a clean cloth cotton to remove any dirt stuck to the faucet mouth. Open the faucet and let the water run for 2 until 5 minutes. Disinfect for 1 minute with a blowtorch or wet cotton with alcohol and then ignite. So note the burning method can only be done on pipe made of metal only. This for pipe made of plastic, disinfect by applying 70% alcohol only. Do not burn. So, and then open the faucet and let the water flow for a moderate rate for 1 until 2 minutes. Do not change the water flow rate after it is set. So, and then next, we open the sterile tail bag, drain the tail bag under the tap water to the 100 ml line. So make sure the water is filled no more than 100 ml and the tail bag does not leak. So turn 3 times and not. Make sure there is some air space for easy shaking during inoculation in the laboratory. And then we put the tail bag that has been built that has been filled into the cool box in vertical order. So make sure the ice pack has been frozen first and the temperature in the cool box is between 1 until 8 degrees Celsius during delivery to the chemical department laboratory. Make sure all samples reach the laboratory within 24 hours. Separate raw water and treated water samples to avoid cross contamination. So this is how we place the sample bag, which is in vertical position in the rack. So this is the proper sampling technique for contamination prevention. We need to prevent pollution from contaminated sampling equipment. So avoid water entering the container by touching the edge and fill the container with gentle water flow to avoid turbulence and air bubbles. For sample identification, we need to enter code 02 for raw water samples and enter code 13 for treated water samples and properly label all container and form. So this is the chemical sampling procedure. We have pre-sampling, on-site sampling, storage and transport. So this is same with the microbe one. Identify and confirm sampling site based on the command schedule, and then prepare sampling bottles according to parameters to be tested, and label each bottle with sampling ID, location, date, and time. So on site sampling step, same also, we flush the tap or source for 2 3 minutes to clear stagnant water, collect sample directly, and filter if needed using 4.5 filter before acid preservation. And then we preserve the sample immediately if required. And then for the storage and transport, store sample in approximately 4 degrees Celsius in the cooler box. Then we transport to laboratory within 24 hours and maintain proper chain of custody record. Okay. Next, uh, for the water sampling chemical analysis, we need to prepare enough water sample bottles with the number of sampling station and also the number of chemical parameter to be analyzed. So use bottle, preservative, and colored label that has have been set. Use a new and clean bottle to avoid contamination. Do not use recycled bottles. Use the correct water sample bottle according to the type of chemical parameter to be analyzed. For example, use bottle A for metal and toughness parameter. So, sample of organic matter. We need to rinse the bottle only once. This is to avoid accumulation of the concentrated of the compound on the walls of the container through absorption. So, volatile organic compound NTHMS. The bottle should be slightly filled and filled slowly to avoid bubbling. So, fill the sample with water until it is full. Make sure there is no air space above so that the water sample does not shake during delivery, especially post water samples for the analysis of organic and volatile organic parameters. It also prevents change in the carbon dioxide, pH, and color due to the oxidation of dissolved gases, precipitates, and so on. So make sure the sample has pH below 2 after acid drop and attach the correct color label to the water sample bottles. Fill in the necessary information on the color label and put the water sample bottles from one sampling station into the same plastic bag before putting them in the cool box. So this is the little bit of water samples, bacteria and also chemical to the laboratory. Bacteria sample should reach the laboratory in less 24 hours. The sample bag is vertical order inside the cool box. Separate raw water sample from treated water contamination to prevent contamination during water sample transportation. Ensure the transport temperature of the bacterial water sample at a cool temperature below 8 degrees Celsius but without freezing. Use crushed uh, ice or ice pack that have been frozen beforehand. So frozen water samples will be rejected by the chemistry department. So prepare a temperature and temperature reference bottle in the cool box and make sure the tire bag does not break in the cool box. Tire bag that break will be rejected by the chemistry department. Okay. So we need to send the sample directly at the lab as soon as possible, preferably on the same day. We need to close sample bottle tightly and the water sample that sent in a cool box filled with sufficient ice pack. For sample that cannot be sent on the same day, keep them on the refrigerator. Delivery careful to avoid broken glass bottles, excessive shaking, or shaking of the water sample containers. For documentation and quality control, for documentation, we need to complete the CAMAM to chemical sampling form for each sample. Record all field data. This is including the site condition and sample temperature. Maintain site chain of custody if samples are handled by multiple personnel. So for quality control measure, use pre-clean and certified contaminant-free bottles. Follow MOH guidelines for volume, preservation, and container type. Conduct periodic training for sampling staff, and also use field blanks or duplicate sample as part of QA or QC. Perform routine audits of sampling practice. So that's all from me. Thank you.